In this presentation, we'll finish looking at how mitochondria generate ATP using the high-energy electron carriers NADH and FADH2. In a previous presentation, we learned about how glucose is broken up step by step to create a number of ATP molecules and several high-energy, reduced, electron-carrying molecules NADH and FADH2. The next step, the electron transport chain, occurs on the inner membrane of the mitochondria. In the next step, NADH and FADH2 deliver their high-energy electrons to a series of proteins embedded in the inner membrane. These proteins, shown here in purple, are arranged into large complexes, and they use the energy of the electrons in order to pump protons from the matrix of the mitochondria into the intermembrane space. We'll look at this in more detail in a minute, but I want you to remember that the goal of electron transport chain is simply to pump protons into that space, making the intermembrane space electrochemically very different than the matrix. So let's look in more detail at the electron transport chain. At the front end, NADH delivers its high-energy electrons to the molecule FMN. FMN accepts the electron because it's more electronegative than NADH. And in the process, NADH becomes oxidized. That is, it loses its electron and becomes NAD+. By accepting the electron, FMN becomes temporarily reduced, and it passes the electron on to the next protein in the chain, which is slightly more electronegative, at which point it returns to its oxidized form. The electron transport chain continues like this, with each subsequent molecule being reduced when it accepts an electron, and then being oxidized when it gives the electron up to the adjacent molecule, which is more electronegative. Notice here that FADH2 dumps its electrons into protein complex 2 rather than complex 1 like NADH. FADH2 has slightly less free energy available to it compared to NADH, and so those electrons do less proton pumping. The next thing to notice is that the final electron acceptor is oxygen. We'll have a lot to say about its role here later. For now, just realize that oxygen is very electronegative, and therefore it allows electrons to drain out of the end of the electron transport chain, and, along with the hydrogen ions, forms water. The oxygen used here is the reason why we breathe. Okay, now we know how electrons move through the chain, let's look at some of the details. In this diagram, the path of the electrons is shown in yellow. So you can see that the electrons enter the electron transport chain either via NADH or FADH2. These molecules become oxidized as they drop their electrons into the chain. Protein complexes 1, 3, and 4 are all responsible for pumping proteins. They use the energy of the electrons passing through them in order to pump protein into the intermembrane space. You can see here a molecule called Q, or ubiquinone. This molecule is very hydrophobic, and it's mobile within that phospholipid bilayer. It serves two functions. The first is that it carries electrons from either protein complex 1 or 2 to protein complex 3. The second function is that while it carries electrons to complex 3, it also carries protons from the matrix in yellow to the intermembrane space in orange. Thus, it helps to increase the concentration of protons in the intermembrane space relative to the matrix. When the electrons reach the end of the electron transport chain, they need to go somewhere. That somewhere is to an oxygen molecule, which is reduced to form water. As we'll see in the next presentation, if there's no oxygen present, then the whole electron transport chain backs up, preventing us from producing ATP. Again, this is why we breathe. The formation of water from hydrogen ions and oxygen also uses up some of the protons from inside the matrix. This helps in increasing the proton gradient that the proton pumps establish. Now, remember that the main function of the electron transport chain was to pump protons from the matrix, the mitochondrial matrix, into the intermembrane space. The presence of so many protons in the intermembrane space sets up a strong electrochemical gradient in which the protons try to get back into the matrix. You can think of this like a dam on a river. The protons are all stacked up on one side of the dam and would like to go downhill, but can't because of the dam. The protons can't travel directly through the membrane because the inside of the membrane is highly hydrophobic and ions are very hydrophilic. However, there is a way for the protons to get back through the membrane. This is through a molecule called ATP synthase, and, like its name suggests, synthesizes ATP. ATP synthase is very much like the turbines in a hydroelectric dam. In a dam, the water above the dam has high potential energy, and as it goes through the turbines, it causes the turbines to spin, generating electricity. In the case of mitochondria, the high concentration of protons on one side of the membrane causes what's called proton motive force. This force is essentially the propensity of protons to move back from the intermembrane space back into the matrix, and is generated both because of the concentration of protons as well as the repulsion of those protons with each other. As the protons are pushed through the ATP synthase, it uses that motion, that proton motive force, to generate ATP directly from ADP and inorganic phosphate. Generating ATP in this way is called oxidative phosphorylation. In a previous lesson, we learned about substrate-level phosphorylation, and this is different because there's no substrate as inorganic phosphate is added directly to ADP to form ATP. This process, by which the energy stored as a proton gradient across the membrane is used to do cellular work in the production of ATP, is called chemiosmosis. We'll see it again when we learn about photosynthesis. So before we move on, I want to just summarize how glucose is broken up for energy. Glycolysis occurs in the cytosol of the cell and involves breaking glucose into two pyruvate molecules generating ATP and the electron carrier NADH. The pyruvate is moved into the mitochondrial matrix and is converted to acetyl-CoA with the production of carbon dioxide and more NADH. The acetyl-CoA enters the citric acid cycle and is successfully broken down to generate more carbon dioxide, more ATP, more NADH, and FADH2. The electron carriers transport their electrons to the inner mitochondrial membrane and deliver them to the electron transport chain. As the electrons travel through the electron transport chain, their energy is used to pump protons into the intermembrane space. The electrons finally end up with oxygen and reduce it to water. The proton gradient that was built up in the intermembrane space is used to drive the production of ATP by oxidative phosphorylation. Now, you may notice that in this figure, it says that a maximum yield of 29 molecules of glucose is produced per molecule of 29 molecules of ATP is produced per molecule of glucose. Your textbook, however, gives that figure at either 30 or 32 molecules of ATP. Why is there a difference? And why don't we know for sure? Well, there are essentially three reasons why we don't have a very precise count of the number of ATP produced per molecule of glucose. The first reason is that oxidative phosphorylation and the electron transport chain are not directly coupled together. Biochemists have estimated the number of protons required to go through ATP synthase to produce one molecule of ATP. They estimate that number to be 4, but in order to generate those 4 protons, electrons must move through the electron transport chain. Some electrons enter the electron transport chain from NADH and others from FADH2. Remember that electrons entering via FADH2 have less energy than those entering from NADH, and therefore pump fewer protons. The second reason for an inexact count is that NADH must be brought from the cytosol where it's produced by glycolysis. As those electrons are brought in from the cytosol, they may be passed to NAD+ in the matrix or to FAD, but whichever one is used will cause a different number of protons to be pumped via the electron transport chain. The last reason for an inexact count is that pyruvate needs to be shuttled into the mitochondria from the cytosol. The active transport also uses the proton motive force that builds up in the intermembrane space. However, by using some of this proton motive force, it reduces the available force to generate ATP using oxidative phosphorylation by ATP synthase.